check the quality of the FastQ file, the tools we are going to perform today is FastQC. So QC is a quality control or quality check to check the quality of a FastQ file. So once we have the FastQ file, we need to check the quality that is used in FastQC. Then we need to do pre-processing. There are the different tools is called CutAdapt, FastP, FastX, Primomatic. All the tools are there. We can able to go and explore each tools as much of it's possible to understand it. So FastQC is most widely using tool to assessments of high throughput sequencing data, where we can able to generate a HTML report that is helps to summarize the sequencing quality, all those things. Why quality check is important? Why we are using FastQC? We are using is to determine sequencing errors. We can able to assign identify the overall data quality. We can able to identify the adapted contamination, whether it has or it has how the GC content present. What is it if they have the sequence duplication? That is also we can able to see from the quality control. We can able to represent the over representation sequences, all those things. You guys, I hope you have worked with FastQCs. And what about others? How familiar with FastQC tool? You have worked already with? Already worked with FastQC? Okay, this will be very fast time. Fine, you have worked. We'll explore it today. Okay. To go into a FastQC, we need to give the input file as a, that is good. We need to give input as a FastQ file. So the FastQ file should be a dot FastQ or dot .gz file, and it will provide the format of HTML report or zip file archives or data, all those results it will provide. So why this FastQ? This is how the FastQ HTML report looks like. We will have information or heat specific way. Important because where it is an advantages which is fast and easy to use. It's a platform which is very independent, which is suitable for a single and pattern data, and which is very important of a step process to do an entire NGS workflow, right? Um, to do installation, FastQC, we can able to work using Galaxy, that is a beginner friendly versions. You can able to download in Windows machine also, or we can able to download the FastQC tool, and we can able to download the FastQ file from NCI SRA, and then we can do just input the FastKey file and we can able to identify the quality check. In other way, where we handling a larger amount of data, we need to download in the Linux softwares. That downloading methodology I have had it over here. It does have a codon dependency, so it just need to be installed with codon, where we can have installation with biocodon FastQC. Or if you want to install into Ubuntu machine, so you can able to do sudo apt installation FastQC and perform the analysis. Just once installation is perfect, you need to give FastQC a read one and read two, which is called, it's a parity and data where we are addition. And at the end, it will provide the FastQC information, the quality check, where we have for the read one information, and then we'll get the information of read two. I hope you guys are joined in your laptops or your systems. Can you go and check about in a Google of what is FastQC? You can able to get into a FastQC software. Meanwhile, parallelly, I also share the screen of uh, sorry. Okay, let me share the screen, entire screen of a website. So if you guys are already using it, can you go and check the FastQC? Sorry. It's showing. So if you go and check FastQC, so the first thing, you can able to see the FastQC, a quality control tool for high throughput sequencing, which is from Vismaya Informatics. So they have introduced the tools where you can able to see about the basics of FastQC file. So it is developed with Java. It's a very fast time, right? Okay, no problem. We go slow by slow. We can able to see FastQC, uh, which is built under Java. So the requirement to install is we need to have a Java running, and we can able to download with Picard. We can able to download it. The latest version is available. If you want to give download, we can able to come down. You can able to see the documentation, how we can get into the data, what are the examples of different formats, is all those things. That is, those information has been highlighted over here. I think. Okay. Meanwhile, if you are working with Galaxy, the few have already have worked with Galaxy. If you go Galaxy, so you can just log in Galaxy, that you guys know, right? You just log in Galaxy and yeah, thank you for sharing. You just log in Galaxy, you will have the homepage of Galaxy. In the tools, you have explored Galaxy already yesterday. It's yes or no? Or I need to give an exploration to how to work with Galaxy. Oh, okay, fine. We'll explore this and then we'll fine. No problem. Uh, this is a Galaxy site. It's built specifically for bioinformatics analysis. Just I'll share. If possible, if any of you can share the link, or this is just search by Galaxy. You can search where all the software tools which is related to our informatics all presented over here. And this is beginner friendly tool to perform the analysis. Where in there, what are the different types of bioinformatics tools, softwares you require to perform, all will be there here. So you can able to see, we can able to download, upload data from here. Thank you for sharing. You can able to download, upload the data over here. Tools associated into a tab, we a different set of tools, like from getting the data and what are the different genomic tools, data sets you have. You can able to FastQ quality control, different types of file formats, different versions is all will be highlighted over here. What type of analysis you can able to perform, RNA-seq, alignment, evolutions, protein folding, statistical analysis, all will be there. As I told, the tools associated, which is also has been check-in here we can analysis. So as the you have explored NCBI SRA, you can identify the data from that. Do you know how to NCBI SRA data, which is directly in Galaxy? So for example, we are going to perform the quality check using FastQC. Later, yeah. At the end of the session, I'll share the slide, or I'll make you to share. That's okay. no problem. You can get to the slide over there. Uh, the question is, for example, we are going to perform a quality check using a FastQ file. How we can get the quality, sorry, from which source we'll get the quality score data? Sorry, FastQ file data. We need to go NCBI SRA. Yes, right. If we go NCBI SRA, I just want E. coli data. Okay, I'm just mentioning E. coli, comma, illumina. I'm just mentioning these two things. I'm not doing anything. I'm just entered it. We can get information which is related to the entire data. So you can see how many 6 lakhs plus data which is related to a specific Illumina E. coli information has been highlighted over here. I'm randomly selecting one data. It's not another selecting it. You can it is from the Illumina. This is the RRID, ERRID. That is also fine. Which is Illumina Nova 6000 end sequencing data, which is from a Illumina, which is submitted from university, which is organism E. coli. You can able to see the size of the data, which is almost 3 GB base for 1,000 informations of MB size of data here. And if you select the run, if you select the run over here, I hope you guys have explored all this yesterday. I think the session will go more into it. So the data is very new. It's not released yet. So we'll go and some other, which is little less data. We'll go with Illumina. Okay, this is a WGS data, which is of less size. Randomly picking any one data, which is of smaller size. So you can able to see. Okay, you can able to see this is the run ID. I hope this information is highlighted. If this is not released, we need to pick and some other data. So this is also not at risk released. Fine. Okay. Randomly, I'll go with other data SARS in SARS-CoV-2. It's a COVID sample. I'll go with SARS-CoV-2 Illumina WGS data. I just want to pick a FastQC file. For that, I'm just trying to get you a very smaller amount of data to perform into a very quick way. So that I'm trying that max. So you can able to see something which is of a COVID-19 wastewater WGS, that is whole genome sequencing data, which is from Illumina MySeq. It's also a sequencing methodology, which is a sequencing length of very less size. It is 151.4 MB data, which is a viral, which is one of the form of viral RNA sequencing data. This is the SRR ID. I'm whether this ID is published or not. If this is also not published, then we are difficult to get it. So you get it information over here. You can get information of metadata, analysis, reads, bases, and you can able to see the FastQ file over here. So this is what we have done yesterday, right? If you want a FASTA file, you can download a FASTA file. If you want a FastQ file, you can download a FastQC. That's what we have. You have seen from our yesterday's session. Is it right? Then yesterday. Okay. So okay, that's fine. I understand. This is how you need to check the data. So let's go. I've just selected one sample, is the sample ID, which is the meta information of that. And in download is this called SRR. This is called ID. It's also called as a sequence ID or accession ID. You can name it anything. If you select this, you can see the information of all over here. Meta information analysis, read information, how the reads looks like. That is also we are going to see from here. So this is how the read looks like, and this is what type of format is it is. Either it is a FASTQ file or a FASTA file. You know the difference between both, right? What is a FASTQ? What is okay? This is absolutely a FASTA file because it starts with a greater than symbol. So this is how the read information of the particular sample looks like. What is the difference between a FASTA and FASTQ? Which is I want a quick. Yeah, FASTQ is a quality score. Yes, awesome. I have a more detail to explain. I don't think so. The time is enough. I hope if it's go beyond the expanded time, is that fine for you guys? Because I want to explain you a little more deeper as you are beginner. Thank you for that. Okay, thank you. That's so good. So if you want to download a FASTA file or a FASTQ file, you just need to mention a FASTA. The downloading it's getting started over here. You can able to see download over here. But I don't want to download here. I'm just passing it or I'm canceling it. It's not happening. Okay. What I need to do for now is I want the data in Galaxy software. So in the Galaxy, we can able to get a directly a FASTQ file. See, the data is downloaded over here. The FASTA file is downloaded. So if you want similarly a FASTQ file, it is also download. We can able to download that. Why this is fastly downloaded? Because the sample size is very, very less. So that sample we can able to download. If the sample size is big or it's more than 5 MB, we can't able to directly download it from the Windows machine. For that, we need to go with Linux softwares or we can use Galaxy to download it. In another easier way to perform the FASTQ analysis, I am taking this sample, the SRR ID or accession ID. I'm just copy it from here. Okay, listen, I'm just copying from here. I'm going into a Galaxy where in the tool menu, what I'm going to search is I want NCBI SRA. What I did, I go into a Galaxy. In the tool menu, what I did the search and see SRA. So there are three options out there. It's very easy to handle. So the latest version of Galaxy is 3.1.1 plus Galaxy. So the tool format is input type. What type of input we are going to give is SRR accession ID. So what I copied the accession ID from SRR, right? I'm going to put this accession ID over here. Okay. and just pasted the accession over here. And what type of input I need to get? I need to get input in a zip compressed FastQ file format. Or if you want to get uncompressed file, you can mention the uncompressed file. Or if you want to go with bzipped file, you can get the bzipped file format. Whichever you want record, you can use it. I'm using this. I'm not doing any changes. I'm just running here. It started running. You can able to see the history over here, where you can able to see the data what we want to do analysis that has been downloaded in our history in our particular login. While it is processing, it is in this brown color. While it is done the processing, it will turn into green and it will provide the results. Or if it is failed, it will turn into a red that the process is not properly. And this is how we'll start working with the Galaxy. This is that much easy to perform analysis. So we don't require to download into our local system. In the Galaxy, almost we can work with 50 to 50 GB of data to perform the analysis. This is how you get the FastQ file over here. Can you get it? The process? Everyone understood this? That's good. Let it take time to. Thank you. That's good. Nice. Okay. Once the data is loaded over here, what we need to do, I have told the main thing to do with the FastQ file is the process of what we're going to learn is what we are going to do. The first step to do QC. Yes, we are going to use the tool do QC is what tools we are going to use it right now? Yep, it's a FastQC tool. I'm not doing anything. Let it process it somewhere here. But I have already uploaded few datas. Uh, you can able to see the specific data which is I already uploaded over here. I will teach you an example. Meanwhile, if this processing has been get over, we use this also and then we use the analysis. Okay. In to do, I go search thing. You can able to see tool a fast QC a read quality report. If you select that, you can able to give information about the tool parameters with analysis. How would the raw data, how does the input data, whether we have any adapters, what are the information, all those things has been highlighted. You can able to see the purpose of FastQC, why it is FastQC, what is the information, how the Galaxy was wrapped with the information, what is the version, all those will be mentioned over here. It is taking little more time to download it over here. Okay. Meanwhile, we'll go here. I have already uploaded the few FastQ file of I have previously performed it. You can able to see something is called par end data. So selecting this, you can able to see there are SRE 38385 thing. If you select this, you can able to see two set of data, forward stand and the reverse stand. This is called par end data. If you want to see, we can able to see information like loaded this code I take. No, I have used this data for example. So what is the accession number? All those information has been highlighted over here. In the previous section, you can able to see the FastQ file. So this is a large file. It is more than 100 kb. You can get the information of everything highlighted here. It starts with at symbol, so it followed by SRRID, then the reader information, then it is followed by the sequences, then it starts with plus symbol, and then it is having what is called actually ASCII characters, that is called fetch for value information. All will be there. That's how the FASTQ file looks like. So we need to do the quality check with the particular sample. So we'll go back to the history. It's still loading over here because of the internet, maybe sometimes. So I'm going into search. If you see, if you want to get into information, I'm just giving a search. You can able to see that that will be empty for you, but I have uploaded the larger file over here. I'm going and selecting a specific data. So something which is called a data over here. Uh, I have selected one data, which is SRR ID of this. I'm just selecting the forward stand. I'm not doing anything. I have selected the forward stand. I'm just running a FastQC quality check. And you can able to see the quality check processing will be taking place over here. Once the quality check has been done, it will provide the web page HTML report. Another one is giving the raw data output as an output. Two information it will provide. So far, you guys can able to get an idea, right? Or it is very difficult to understand how it looks, or it's how what you guys are feeling. The quality check processes gets over, so you can able to see the history. You have two information. One is raw data information, another one is a web information. The first thing we're going to see is a web information. So this is how the report, the FastQC report looks like. You can able to see the entire results. I'm going to explain you from the step by step into a little more detail. So that's how the report looks like. So you can able to see. Thank you. You can able to see information of the report, which is having basic statistics, which is telling about what is the file format, what is the file name, how it is encoding, what is the total sequence length, what type of sequencing takes place, what is total bases they have, how the sequencing flags, whether it is in a good quality or bad quality, what is the sequence length, what is the percentage of this GC content, all those information it have. And you can have a color changes of this. I'll explain everything in a slide because the explanation which is little more into deeper. You can have a different graphical representation for the each thing. You can able to see a green mark and into mark and the warning mark. Each symbolize each specific thing. On all this highlighted summary things, which is very, very important to understand, to process the quality of a particular data. So let me explain that in every graph what are things are there, and this is how the quality reports looks like. Similarly, if you want to do a quality check for the reverse band, the same process, we need to go into our tools. We need to search FastQC. We go into check FastQC and do the same process. We need to select with, so this is how our data is not uploaded yet. I think so. And we need to go back. We need to upload the reverse data. So we have uploaded the reverse data, right? So we have worked with the parity and data. We need to check the reverse end data. I wrongly mentioned it. Okay. We need to perform the analysis. I have a question over here. If you have your data FASTQ file in your local machine. What will you do with the Galaxy? Do you have an idea about it? Oh my God. I need more time. Okay, is it okay if the session goes for another half an hour? I'll try to widen up as much as possible. We should upload from the local machine. Yes, yes, absolutely yes. So here is an upload option. So you just need to choose from the local machine. So once we have uploaded the data, so in my download, they have a factor data here. If you want to upload, you just need to select, open it, and you need to upload it. That's it. Your data get uploaded, and you can able to have in your login, and we can able to go on analysis. That's it. Well, you can able to do that, right? Now we can able to see what the FastQC report tells us. Let be understood so far to everyone. Good. Nice. Okay, let us go and see what the quality reports is telling you. One second. Um, okay. So this is how the we have see the report, right? The summary of report. The report contains multiple models over there. So it just have information about basic statistics, per base quality score, per type quality score, per sequence quality score, per base content, per GC content. All these information will be there in the report. These are the information. Someone asked in between what determine the quality scores, right? This all determines the quality scores. This all very much important before performing the mathematical analysis. So if it is a green tick mark, there is no issue in deduction. If it's a warning symbol, it means a minor concern which is there. If it is X symbol, the significant issue is it's there. We need to have a proper attention before we need to perform the analysis. Okay. So all the information it holds, right? So first thing, just have a basic information that is all the basic statistics, which is telling information about these data, what type of data, what are the sequence length, what is the matrices, how the GC content, all will be information will be there. And the next thing is called per base sequence quality. So this displays the quality scores distributions across the different nucleotide position. So the x-axis is the positions of a reads in base pairs, and the y-axis represents the quality scores. And you can able to see the three different layers of colors, which is of a red region, yellow region, or orange regions, or a green regions. The red regions, the quality is very poor. Orange region is acceptable. If the bar plots, which is all in the green regions, it's excellent. The quality score is above 28%. So we can consider that sample to perform the analysis. So that is in the green color. So it is often to be considered from the 3 ends of Illumina reads to understand whether the signaling has been taken place properly. If it's see, you can able to see the graph. It is completely in the green regions where it tells all these qualities are good. We can able to consider this for the analysis. Then coming up the second thing is called per sequence quality score. So it shall be in the language in a peak. The peak should be in a range of 30 to 40. Where if it is 30 to 40, the average quality is good. So where you can able to see the peak is exactly at 30, the quality is very good. We can consider this data for our analysis. The sequence level quality is absolutely good. The next thing is per base sequence content, where it just helps to understand the percentage of content, how the nucleotides positions are. So whatever the pattern is, if the pattern is, you can able to see the pattern should be in a straight line. If it is deviated due to a error in the library preparation, or maybe due to primer based errors, or it may be due to contaminations of happened during the process of doing analysis. It's all the reasons of what it is has been taken place. We need to consider this, and we need to do a trimming process, because adapter contamination is there. But these are the things which is helps to do a pre-processing step, that is what's trimming and filtering. That is what in continue we are going to see that. Our next thing is called a GC content analysis, where we can able to understand what is the percentage of a GC content is across the organisms. So if it is E. coli, it should be approximately equivalent to 50%. It is Mycema bacterium, it is 65%. It's a standard thing. It can be equivalent. It is little below or little high, it's fine. If it is too below or too high, that might be a deviation, mixed culture happen taken places, or may happen with the sequencing artifacts or all those things. So if it is a normal, it should be in the percentage of the standard condition it should has. Next thing is called sequence duplication graph, which just tells you how often the ideal reads occurs. The sequence duplication is occurred in the sequences maybe because of PCR amplification methodology, or maybe because of high sequencing depth, or it may be due to low library complexity. In all those conditions, the sequence duplication might be happen. So if it is lower the duplication, they preferred for genomic analysis. If it is higher the duplication, it may be due to a PCR biases or any types of expected error happened during that time. Overrepresentation and the adapted contamination. So adapted contamination, it's based upon the synthetic DNA fragments added during a library preparation process. So if it is the sequence, the sample has the overrepresentation content or a adapted contamination, we need to remove during a trimming process, and then we need to do a filtering process. That's where the adaptive trimming all important before going into a library preparation. Sorry, before going into a downstream analysis. That is where I have mentioned over here. You can able to see in the graph, you can able to see the overrepresentation of these sequences, where it is due to adapted contamination, PCR primers, or ribosomal RNA, or contamination agent. So all this leads to do perform the uh, trimming and filtering process. That's where the pre-processing comes in.